Introduction to drugs acting on the central nervous system. Neuroanatomy and neurophysiology.
The central nervous system is composed of brain and spinal cord. The functional unit of the nervous system is called the nerve cell or the neuron. The receiving end of the neuron is called the dendritic region and the transmitting end are called the axonal terminals. Nerve impulses are transmitted in two types of synapses. First is the electrical synapse. It is characterized by channels that conduct impulses. Second is the chemically mediated synapse. It always transmits signals. Physiological anatomy of synapses. Synaptic cleft separates the presynaptic terminal and the postsynaptic neural stroma. Presynaptic terminal is the location of transmitter vesicles. It contains transmitter chemicals that excite or inhibit postsynaptic neural functions. Action potential initiates the release of transmitters by the help of the voltage-gated calcium channels. The amount of transmitter released is directly related to the number of calcium ions that enters the terminal. Receptor proteins have two important components. First is the binding component. Second is the ionophore component. So there are two types of ionophores. First is the ion channels, provides an aqueous route, cationic channels which allows the passage of sodium ions, and anionic channels which opens the chloride channels and inhibit neural function. Second is the second messenger activator. Second messenger to alter cell functions. Neurotransmitters are usually classified into two major groups. One of the major groups in the neurotransmitter is the small molecule, rapidly acting transmitters, synthesized in the cytosol of the presynaptic terminal and causes most of the responses in the CNS. After their actions on the receptors, they are either destroyed locally by enzymes, diffuse out of the cleft, or are absorbed by the active transport back into the transmitter vesicles. Some important small molecule, rapidly acting neurotransmitters. Acetylcholine. The primary effect is to close potassium, calcium, or chloride channels, leading to depolarization or hypolarization. It is widely distributed throughout the central nervous system. It is inactivated via hydrolysis. Norepinephrine. It is important in the control of sleep and wakefulness, mood and emotional behavior, and temperature, and it activates excitatory receptors. Epinephrine, also known as adrenaline, in the reticular formation of the medulla. Dopamine. It is the major neurotransmitter in the central nervous system, and it controls movement, disturbance of behavior, and the hypothalamic pituitary endocrine system. Serotonin, or 5-HT, inhibits pain pathway in the spinal cord and helps control behavioral mood. Central nervous system's regulatory function, sleep and wakefulness, mood and emotion, temperature, appetite, and neuroendocrine control. Present in highest concentration in blood platelets and gastrointestinal tract. Histamine, mostly located in the posterior hypothalamus. Regulation of arousal, temperature, and vascular dynamics. Amino acids, highest concentration in the central nervous system. GABA major inhibitory transmitter receptor in the central nervous system. Greatest concentration in basal ganglia, hippocampus, cerebellum, and hypothalamus in the brain and in the substantia gelatinosa of the dorsal horn of the spinal cord. Glycine, inhibiting transmitter between spinal interneurons and motor neurons, and increases conductance of chloride. Action restricted to spinal cord, lower brainstem, and retina. Glutamate and aspirate. High concentration in the brain. The primary excitatory transmitter in the brain and the spinal cord. Glutamate receptors, G protein-coupled receptors, and ligand-gated ion channel receptors. Ion-gated receptors, kainate receptor and AMPA receptor. Nitric oxide, responsible for long-term behavior and memory, almost instantly synthesized and does not alter membrane potential. Second major neurotransmitter, neuropeptides, present in smaller quantities but are far more potent. Synthesis directed by messenger RNA by ribosomes. It is packed in the smooth ER and transported to the nerve terminal for later release by a calcium-dependent process. In many cases, the neuropeptides are present in the same terminal as the small molecule neurotransmitter and are released together with them. Peptides are not recycled but inactivated by enzymatic breakdown. Substance P can be found in the brain and intestine. Primary effect in the brain is an excitatory action on dopaminergic neurons, prolonged duration of action, and the release by primary afferent nerve fibers in the dorsal horn of the spinal cord and mediate sensation of the noxious stimuli. Opioid peptides have agonist activity at opiate receptors. Endorphin transmitter at a number of synapses in the pain modularity mechanism. Three branches. First is the pro-opiomelanocortin-derived peptide, an interior and intermediate lobes of pituitary gland modulating effect. Second is the proenkaphalin-derived peptide. And the third is the prodynorphin-derived peptide, widely distributed throughout the central nervous system. Enkaphalins and beta-endorphins, involved in the mediating analgesia. Subtypes of opiate receptors. First is the mu receptor site, at which analgesia is mediated in the brain. Agonists include morphine and beta-endorphin. Stimulation of the receptor is associated with the respiratory depression, dependence, meiosis, and decrease in gastrointestinal propulsive motility. Second is the delta receptor strongly influenced by leukoenkaphalins, analgesia-mediated behavioral and sedative actions. Third is the kappa receptor, by dynorphin and mediated spinal anglesia, but also causes dysophoria. Action of drug in the central nervous system. Drug action in the central nervous system may be specific or nonspecific. Specifically, effects are produced by modifying some steps or mechanism in the chemical synaptic transmission of nerve impulses. Nerve impulses conduction can be prevented by local anesthetic to nerve fiber. Synaptic transmission can be blocked or depressed at the presynaptic site by halting transmitter synthesis or storage. Other drugs increase the release of transmitters, may affect the reuptake or the degradation of drugs once they are released into the synaptic cleft. Drug may also act on the postsynaptic membrane like neurotransmitter on the receptors, or block receptor function at the level of the receptor or affect the further transduction of the signal from the receptor. Some drugs are nonspecific, like inhaled drugs anesthetic. Blood-brain barrier. The boundary is described as between blood and tissue of the central nervous system that is less permeable to large, water-soluble, and/or ionic molecules. Limits penetration into the brain of the hydrophilic substance like dissociated acid, bases, and proteins. Exits both in the brain tissue capillaries and in the choroid plexus, and is created by the uniquely tight junction between the capillary endothelial cells or the epithelial cells of the choroid plexus and by the close association to the capillary walls of the glial cells. However, water, carbon dioxide, oxygen, and most lipid-soluble substance, like anesthetic, freely pass through the cell membrane and gain entrance to the brain. A carrier-mediated transport mechanism to transfer micronutrients such as vitamins, macronutrients such as glucose, amino acids, ions, and certain other substances. System included active transport, facilitated diffusion, and endosomal transport.